And since the cardiovascular system has quite a lot of components to it, like we said, it separates into at least a couple different chapters. Chapter 12, which is the blood, and Chapter 13, which is the rest of the cardiovascular system, specifically focusing on the heart, but then the vascular part has to do with the blood vessels as well. So with the blood chapter, what we're going to be looking at is, on a certain level, the blood, you think, oh, wow, that's just, whatever, that's what's inside of it, and that's not that important. But we're going to find that there are multiple different parts of the blood that are going to be important, both the cells as well as the fluids, and they're going to be supporting all these different organ systems that you learned about previously and that you'll learn about still. Alright, so when we look at the blood, it actually counts as what we call a connective tissue. Now, way back when, we talked about different types of tissues in the body. You spent a little bit of time learning about epithelial tissues, but we said there are three other tissue types, right? There's epithelial, that's the one you looked in detail at, the covering and lining. There's muscle, there's nervous, and then there's connective tissue. Now, most connective tissues are the ones you might think of, like bone and cartilage, etc. But blood also counts as a connective tissue because by definition our different connective tissues will have a bunch of cells sitting within an extracellular matrix. So they're not necessarily right packed up one against each other, but they're sitting in some type of an extracellular matrix. In this case, a fluid that we call the plasma. So even though it doesn't have a function in terms of connection, if you will, it does resemble anatomically our other types of connective tissues. Much like the bone, we see those osteocytes sitting in the mineral matrix or whatever. So when we look at the blood, though, like we said, there's going to be lots of different components to it. Of course, there are going to be the cells. These are going to be red and white blood cells. But there's also going to be these cellular fragments. These are not entire cells. They are pieces of cells that we call platelets. And so when we talk about the kind of, if you will, hard parts inside of blood, we don't always just say cells. We'll say cellular components or cellular elements because, as we mentioned, these platelets are not, in fact, whole cells. But they form what we call these cellular elements or formed elements, the quote-unquote hard parts of the blood. Now, of course, in addition to that, we said we have the plasma. So the plasma is going to have some roles of its own. It's not just there to float the red and the white blood cells and the platelets, but it has many functions on its own. So overall, though, the blood is there for transport. The cardiovascular system is about transporting this blood around, which is going to transport substances through the body and help to maintain this stable internal environment. It's going to help to maintain homeostasis in many different ways. It's going to help to maintain heat in the body. It's going to help to maintain pH, nutrient levels, waste levels, things like that. Alright, so we know that there's going to be these formed elements, or kind of the solid part, or the cellular part, and then there's going to be this fluid. What's the balance? How much of each do we have? Typically, in a blood sample, it's normally about 45% cells. Now, this is going to vary depending on whether you're male or female, depending on whether you live at high altitudes or not, depending on maybe your body size or whether you are dehydrated or anything like that. But on average, it's about 45% cells and about 55% plasma. So it's slightly more than half fluid and then slightly less than half cells. And you see this term every once in a while, right? It's called hematocrit. Hematocrit is basically the packed cell volume, specifically the red blood cells that we find inside of a tube of blood. So if you take a sample of blood, what they usually do is they centrifuge it, they spin it, and as they spin it, the cellular components are going to separate from the fluid component. And so down at the bottom, the heavier cells are going to end up down there, and the packed red cell volume is going to be what we call hematocrit, and that's about 45%. We'll find that there are, in terms of red and white blood cells and platelets, there are so many more red blood cells compared to the white blood cells or the platelets that most of your cell volume is dedicated to red blood cells, and there's only a very thin layer on top of that, less than 1% of the total volume, that is white blood cells or platelets. But in any case, cellular components 45% normally, 55% plasma. And the plasma is going to be mostly water, but it's also going to have a lot of other small molecular components, vitamins, nutrients, waste products, gases, ions or electrolytes, all kinds of things in there. So this figure here is a great figure to show us a lot of what we're talking about in terms of what goes and makes up the blood. Okay? So again, on the left side here we see 55% plasma, 45% cellular elements. When we look at the plasma, here we can see a list of all these different molecules and ions found in it. Now obviously, mostly water, right? 95% or more water. That's the solvent that carry all these other substances like ions. And you look at this list: sodium, potassium, calcium, magnesium, chloride, bicarbonate. This is just a handful of ions that we can find inside of the blood. We know that sodium and potassium are very important in terms of action potentials to allow neurons to transmit their signals. It's also important in terms of the muscles because they generate an action potential as well. Calcium is very important in terms of the muscles, right? It is the ion that allows the actin and the myosin to interact. Magnesium, chloride, a lot of these maintain an electrical balance in the body or act as cofactors to allow enzymes to work, etc. And bicarbonate, interestingly, we'll talk about it with respiratory system as well, but bicarbonate, the bicarbonate ion, is the way that most carbon dioxide is actually transported in the blood. Other molecules, plasma proteins. Proteins, right? Remember, proteins are basically a very long string of amino acids linked together. And there are three major proteins that we find in the plasma. One is called albumin. It works to maintain what's called osmotic balance, meaning that we want to make sure that the fluid balance between your tissues and the blood is at the right levels. When you get dehydrated, your cells get smaller, they shrink, and in order to get out of that situation, you're going to drink fluids. As you drink fluids, that's going to be absorbed in your digestive system and go into the blood. And then from the blood, we want to deliver it out to the tissues. But even though, we think about a lot of times the blood just delivering things out to the tissues, you really have to remember that the blood is more like your postal worker or your mail carrier. Your blood works to deliver and significantly to pick up from the tissues as well, whether it's pick up waste products like carbon dioxide and different types of other metabolic waste products, or fluids, because we want to get, like we said, have a fluid balance between our tissues and the blood. And the albumin helps to draw fluids back into the blood from the tissues as necessary. We have another protein called fibrinogen. We'll talk about its importance at the end of the chapter, and that has to do with clotting. And then there are also these what are called immunoglobulins, which are basically antibodies. Antibodies that help to protect you from various types of the disease-causing agents that we call pathogens. Other things that we find in the blood: nutrients, right, that are absorbed from the digestive system, waste products from metabolism, respiratory gases, oxygen and carbon dioxide. Now technically oxygen is mainly carried by the red blood cells, but there is a small percentage that is actually diffused straight into the plasma, as well as a small percentage of carbon dioxide that is found in the plasma in that form as well. And then lastly, we have hormones. And hormones are going to be, as we saw from endocrine system right before the last exam, hormones are the chemical messengers that communicate from an endocrine gland to its target organ. So really a lot of different functions, we would say, associated with the plasma because it has all these things inside of it. Now when we look to these formed elements, of course these also have important functions. We have the red blood cells known as erythrocytes. Right? Cyte means cell and erythro means red. We have leukocytes, which are the white blood cells. And there are 5 different types of leukocytes, and we will talk about why there are 5 different types. But in essence, remember, there's always this relationship between structure and function. So if we have 5 different types of white blood cells, they probably have 5 different functions. And then lastly, we have the platelets. And the platelets serve in the process of blood clotting. They kind of initiate the process of blood clotting. Now when we look at these, like we said, look at the numbers here. This is per cubic millimeter of blood. So a cubic millimeter of blood, if you were to put your fingers together to where they're almost touching, that's about a millimeter. So a cubic millimeter, a cube, a millimeter on each side of blood, has 5 to 6 million red blood cells in it. It's only got 5 to 10,000 white blood cells and maybe a quarter to a half a million of our platelets. So looking at those numbers, now you get a good idea of why your blood looks red, because it's dominated in terms of numbers by the red blood cells. And again, that's what we're seeing here in the center as well, right? We see a centrifuged blood sample here. All the yellow at the top is the straw-colored plasma. All of this down below is the hematocrit, basically the packed red cell volume. And then there's just a little layer called the buffy coat that sits right on top of the red cells. And that would be the white blood cells and the platelets. Alright, so we're going to talk about our different cellular components first, and then we'll get into the plasma, and then we'll finish up talking about hemostasis and how we keep blood and maintain blood. Alright, so red blood cells, we said they're called erythrocytes. You do need to know the official name as well, of course. They have a very unique cell shape. So rather than the generic cell where we've got, just some sort of generic circle with a dot in it that's like a big nucleus, right? With red blood cells, in fact, these are what are called biconcave discs, meaning they dent in on both sides, on two sides. And these have no nuclei to them, okay? They are basically, they're a very rare anucleate, right, no nucleus cell in the body. Very rare in terms of there's not many cells in the body that don't have a nucleus. And their main function is as big bags of hemoglobin. They carry the molecule hemoglobin, and hemoglobin is very important because it's the hemoglobin that primarily has the function of transporting oxygen. So when you talk about the blood taking oxygen out to the tissues, those oxygen molecules, for the most part, are attached to this iron-containing hemoglobin molecule. And there's all kinds of hemoglobin molecules in each red blood cell. And each hemoglobin molecule can actually hold on to 4 different molecules of oxygen. So these red blood cells are filled with hemoglobin. They have it in the plasma membrane, etc. And those hemoglobin molecules carry oxygen. It's their main function. Now, because the hemoglobin contains iron, when the oxygen combines with the hemoglobin, it produces what's called oxyhemoglobin, and it looks very bright red. And so oxygenated blood, like you think about in arteries or something like that, and we'll talk about that later, that oxygenated blood appears red. But deoxygenated blood actually is called deoxyhemoglobin, and it doesn't appear blue like you think about with the veins or you see it drawn out. It's just a darker kind of muddy red instead of a bright red. And the whole idea there is that with the iron inside of the hemoglobin, much like if you think about metal that's rusting that has iron in it, when it gets oxidized it turns red, right? Rust. And so when the oxygen also is in association with the iron in the hemoglobin, it also looks brightly red. Okay, so like we said, these red blood cells discard their nuclei. They originally are formed like most any other cells, and they're formed in the red bone marrow inside of the bones. It's one of the major functions for the bones besides, support in the body, etc. And before they leave the red bone marrow, they actually discard their organelles and specifically their nucleus. So interestingly, you can imagine these red blood cells don't actually last that long, because they don't have the different organelles to support themselves, to do their metabolism, etc. They don't have a nucleus that is the blueprint for reproduction, and so they can't produce new red blood cells. So they last about 3 or 4 months, and then they basically die. So here's a diagram and a photo of red blood cells. Again, you can see the biconcave disc. You can see where it dents in on either side here, right? And they think that that basically allows these red blood cells to have more surface area. And more surface area means that it can hold more hemoglobin. It also allows these red blood cells to flex or bend in the center, because as these red blood cells are going around the body, they're ultimately going to get to blood vessels called capillaries that are so small that these red blood cells actually have to go through the capillaries single file, and in some cases they kind of have to bend in half in order to get through those capillary beds. Okay, so like we said, our red blood cells come from the red bone marrow. It's contained within the bones in the spongy bone area down towards the epiphyses, right, the ends of the bones, and that's where they are created. Like we said, they generally last about 3 or 4 months, and the red blood cell production is regulated mainly by a hormone that comes from the kidney, and it's called erythropoietin. So there's the erythro part again, meaning red. And erythropoietin comes from the kidney because the kidney, one of the kidney's main function is to regulate the blood. A lot of blood goes through the kidney. If we have too much fluid because we've been drinking a lot, the kidney is going to take out that excess fluid. It's going to take out waste products, etc. And so the kidney, because it's in contact and monitoring the blood every second of the day, it, to a certain extent, makes sense that the kidney would be an organ that would monitor the blood, and if we're getting too low in terms of the red blood cell number or the amount of oxygen that the blood can carry, right, because the red blood cells carry oxygen, then what the kidneys do is they secrete a hormone called erythropoietin that travels through the bloodstream, goes back to the red bone marrow, and tells the red bone marrow to kick up its production of red blood cells. Now ultimately, after it's kicked up its production of red blood cells, then the problem is going to be solved. Your blood's going to be able to carry more oxygen, the issue is going to then go away, and so the kidneys can then stop secreting their erythropoietin. But it's basically this, negative feedback loop where it's monitoring it. Now, if you have blood that has not enough red blood cells, or basically can't carry enough oxygen, it could be not enough cells, it could be the hemoglobin isn't working well or isn't formed right or something like that, like you don't get enough iron in your diet, and so the hemoglobin can't carry the oxygen properly. That state is something that we call anemia. So anemia is where your capacity, your ability to carry oxygen in the blood, for whatever reason, is diminished, is reduced. Now we know that the red blood cells are created in the red bone marrow. They live about 120 days, but what happens when they wear out and die off? What happens to them? Well, in fact, there are macrophages, right? Macro means big, phage means to eat. Macrophages, these are big cells, big eating cells that actually will engulf and get rid of these worn out red blood cells. And these macrophages exist in the liver and in the spleen. And so those worn out red blood cells will get taken out there, chewed up, if you will, and certain components will actually get recycled for us to make new red blood cells as well. So, like we said, it's a dynamic between the red bone marrow, like we see in the bottom of this slide, and to a certain extent the kidney. Now, mostly it's the kidney that puts out the erythropoietin, technically hormones from the liver as well, but it's this negative feedback loop that actually maintains the right amount of red blood cells in the body. Okay, so those are red blood cells. Pretty plain, these are bags that carry hemoglobin, which binds to oxygen. That's their main function. All right, white blood cells. White blood cells are called leukocytes, and they overall have a job of bodily defense. But like we saw, there are 5 different types of leukocytes, so that means they're going to have slightly different functions. They'll all work to protect you in some way, but in slightly different ways. Now, the white blood cells also tend to come from the red bone marrow. Again, the red bone marrow appears red because there's so many red blood cells being produced, but white blood cells come from there as well, and they come from these earlier stem cells that are called hemocytoblasts. Hemo, right, referring to, in essence, referring to blood. All right, so we've got these 5 different types of white blood cells. They're called neutrophils, eosinophils, basophils, monocytes, and lymphocytes. And so when we look at them, here's a table. This is table 12.1. It's a very useful table that shows are different types of cellular components and what they do. I put a copy of this, a JPEG of this on WebCT, I think, as well, but you should be able to find it in your book. So if you look at the columns in this table, you see the first column is the component, the cellular component, gives you a description of it, gives you the relative number present per cubic millimeter, and then gives you a function. Your main concern here, is going to be the first column and the last column. You need to name be able to know the different cellular components of the blood, and then tell me a function for each of them. All right, so we went through red blood cells, but the white blood cells. We know that the white blood cells work in bodily defense to destroy these things called pathogens, right? A pathogen is a disease-causing agent. Now, there's 5 different types of white blood cells, or leukocytes. They're organized into 2 different groups. First, there are the granulocytes. Basically, when you see them in a microscope, you can actually see little granules inside of their cytoplasm. And then there are the agranulocytes, right? A means without, those that don't have granules. Now, the easy way to remember that is if you notice all of the granulocytes end with "phil," okay? And all the agranulocytes end with "cyte." Okay? But you do need to know which are granulocytes, which are agranulocytes. You need to know the names of these 5 different white blood cells. Now let's look at the functions. Neutrophils. If we go over to the last column here, we see neutrophils are basically phagocytes. So they're almost all these cells are bigger than the red blood cells. The neutrophil is about 1.5 times the size of a red blood cell. And so it's not that much bigger, but it's basically going to phagocytize small particles. Maybe fragments of cells or something like that that have ended up in the bloodstream or in the tissues. Eosinophils. We look at eosinophils. Eosinophils are pretty interesting. They work to kill parasites and help control inflammation and allergic reactions. So if you have a tapeworm or something like that, eosinophils will actually be trying to get rid of that. Basophils. Basophils release what's called heparin and histamine. Now, histamine is a molecule that leads or basically causes inflammation. So, you know, if you look at that word, most people are familiar with something that in their everyday life that relates to that. Everybody knows about something like Benadryl or Sudafed, which basically are antihistamines, right? Antihistamines. If you have an allergy and you're all congested in your nose or itchy eyes or something like that, what do you want to do? You want to get rid of those symptoms, so what do you take? An antihistamine. The histamine produces the swelling, like swelling in your nasal cavity, or even if you're having an allergic reaction or something like that. Benadryl can help that. I don't know if you saw, there was a movie Will Smith did called Hitch a while ago, where he would set people up together, and during that movie, he's on a date with this woman, and he eats something that he's allergic to, and so he starts to get swelling, like swelling in his ear and his nose and his whole face, etc. And what do? They go to the drugstore and they get Benadryl. Benadryl, which is basically an antihistamine. Okay, so anyway, it produces swelling. Now, it could be swelling in a variety of different areas, but basophils are going to produce this inflammation or swelling, which is going to allow other bodily defenses to get to the site of infection. Heparin is something else. Heparin is a molecule that it helps prevent blood clots. So we want to form clots when necessary, when you have a broken blood vessel or something like that, but we want to make sure that we don't form blood clots at any other time. So basophils have a couple different functions. Notice then, the eosinophils and the basophils kind of have opposite functions. We see eosinophils help to control inflammation as one of their functions, whereas with the secretion of histamine, the basophils induce or start inflammation. All right, what else do we have there? Monocytes. Monocytes are actually a very large cell. They're 2 to 3 times the size of a red blood cell, and they are phagocytes as well, like the neutrophils, but they're going to phagocytize large particles or cells, like whole bacteria or larger worn-out red blood cells or something like that. And both the neutrophils and the monocytes, like we said, they're phagocytes, and they're actually in the bloodstream like all of these different white blood cells, but in fact you can find these 2 phagocytes often outside of the bloodstream as well. Because if you have an infection, right, if you have an infection, generally it's not going to be an infection in your bloodstream. It's going to be somewhere else, like in your skin or in a particular organ or whatever. So these cells that are part of the bodily defenses have to be able to get to the site of infection. And so the neutrophils and the monocytes, these phagocytes, will actually leave the bloodstream, like at the capillary level. They'll squeeze out between the cells, and they'll move in what we call a amoeboid movement. They'll squeeze out, and they'll move to the site of infection. They'll move kind of like a snail, kind of squishing themselves out and drawing themselves along. So they're pretty interesting. Lastly, we have the lymphocytes, right, or other type of agranulocyte. And at this point, we'll just say they provide immunity. We know immunity is, a specific type of bodily defense, but we have a whole chapter coming up on lymphatic or the immune system, and so we're going to get into the details of that then, but we will, for now, just say that they provide immunity, lymphocytes. Okay? So those are the 5 different types of white blood cells. Now technically, over half, if we look at this number, right, over half of all the white blood cells you have in your body are going to be neutrophils. Those are going to be followed by the lymphocytes, which are very important in terms of what we call the immune system, and those are going to be followed by the monocytes, our other types of phagocytes. So those that have very large important functions, we find more of these white blood cells compared to others. Last thing I want to address while we're still on the slide is, if you look at the bottom there, it says platelets. Platelets are also known as thrombocytes, and platelets, like we said, have a function in terms of blood clotting or coagulation. So we'll talk about them later on, but they also are contained within this table here. And this slide here just kind of gives you some images of the different cells. In the lower right hand corner we have a scanning electron micrograph, which shows basically a capillary, well no, maybe an arterial, a little bit bigger, and we see red blood cells here, right? You can see they're biconcave, you can see them dented in on both sides, and then these other ones are the white blood cells. They're a bit bigger. And if you look in the upper left hand corner here, we see the same thing. Now this is a regular microscope slide, light microscope slide. All of these pinkish cells, kind of brown pinkish cells, are the red blood cells. And notice in the center of these red blood cells, it appears lighter, right? In the very center, that's where it's thinner because it dents in, so it appears lighter. You can see that over here too. The purple cells that you see here are going to be the white blood cells. Now on the one hand you're going, well, I don't get that, they look purple. Well, in light microscopy they often put stains in with the cells so that you can actually see them a bit better. And so oftentimes it's the nucleus of the cell that takes up the stain the best, and so since it's only the white blood cells that have nuclei, they actually stain the most in the purple. Okay? Some of the other things, you can actually see a few platelets as well. See that little spot right there? There's a little spot right over here. There's another little spot up here. Those are platelets. Like we said, platelets are cell fragments. And here we are talking about platelets, right? So we talked about red blood cells, white blood cells, and now platelets. As we said, the platelets are only fragments of cells. When they're formed, also in the red bone marrow, they come from a precursor cell that we call a megakaryocyte. This is a really large cell that has a bunch of different nuclei. And interestingly, the way that these platelets enter the bloodstream is this megakaryocyte comes up to a blood vessel like a capillary. And this capillary is a special type of capillary. It has holes in it, and the capillary works kind of like a cheese grater. And this big cell kind of pushes into it, and as it pushes into it, it slices and dices that megakaryocyte into these small cellular fragments that we call platelets. And then those fragments are floating around in the bloodstream to help repair damaged blood vessels. All right, and at the very end of the lecture, we'll talk about that hemostasis or the clotting process again. But we'll leave that for now in terms of the functions of our different cells. Okay, so now we get to the other component of the blood, the plasma. We said that the plasma is basically the fluid part of the blood. It's mostly water, but it has a bunch of other substances in it. So it's going to be transporting a lot of other substances that we normally, a lot of people commonly think, well, that must be with the white blood cells, the red blood cells, or something like that. They must do that important function. But a lot of these functions are just with the plasma itself. So things that we can find in the plasma. We said plasma proteins. They're the most abundant molecules, large molecules that we find inside of the bloodstream. We said there are the albumins that help with the osmotic pressure of the blood, making sure that there is water balance or fluid balance between the blood and the tissues. The globulins, which we said also are known as immunoglobulins, or basically antibodies. Mostly they have immune functions. They work to protect you against various bacteria or viruses or something like that. But some of them also transport vitamins and lipids. And then lastly, we have a protein that's called fibrinogen. And it has a very critical role in blood coagulation or clotting. In its inactive form, when it's floating around, we call it fibrinogen. But when it gets activated, we take off this OGN, and now it's just called fibrin. And when it's in the fibrin form, it actually forms, what do you know? Fibers. These protein fibers that work like a lattice work, like a spider web, to reinforce the clot and to stabilize it and make sure that it's there as long as the blood vessel needs it. Okay, what other things do we find in the plasma? Nutrients and gases. So gases are basically oxygen and carbon dioxide. Now we say that oxygen is mainly transported associated with hemoglobin, while carbon dioxide is basically mostly transported in the plasma in the form of bicarbonate. But we still do get some of each in the plasma itself, in their intact forms, oxygen and carbon dioxide. Nutrients that you get from the food that you eat are also transported via the blood to the different target cells that need them. Amino acids are the subunits or the building blocks of proteins. Monosaccharides are the simple sugars that come from carbohydrates. Nucleotides come from nucleic acids from chopping up or eating up DNA from cells, plant or animal cells. And then lipids technically they go down to what are called monoglycerides and fatty acids. So when they break up, technically you can transport some lipids in their larger forms as well. But these are all going to be transported in the blood. So it has an important function in terms of transports of nutrients. Transports of waste products. You have waste products that are coming from metabolism, things like urea and uric acid. Those are things that are going to be produced by the cell during its metabolic activities. We want to get rid of them before they become toxic to the cell. Electrolytes. Electrolytes are basically ions. Now these are going to be used, like we said, by certain cells, for instance like the neurons and the muscle cells, or other types of active cells in terms of balancing their concentrations of ions or their electrical gradient across their membrane. And so sodium, potassium, magnesium, calcium, these are all floating around in the plasma. And so like we said, these are basically ions. They're ions that come from salts. And interestingly, you know, when you look at this word electrolytes, you hear about this a lot in different types of commercials. So if you think about electrolytes, where you might have heard that in a commercial, like in different sports drinks like Gatorade. So they talk about how, you know, you're replenishing fluids and electrolytes. You know, a lot of people, they look at that and they're thinking, whoa, electrolytes, I don't know what that is, but obviously I need them because they like leak out of my body in bright orange or blue or something like that when I'm sweating. Well, like we've been talking about today, electrolytes are basically salts, ions from salts. So, you know, if you want to make yourself some Gatorade, right, without the color and sugar and stuff like that, you take some water, you put some salt in it, now you have your generic form of Gatorade. So it's not like anything special like pixie dust or anything, right? These electrolytes are basically ions from salts. Obviously you do need them. We said this, lots of cells need them for different reasons, but it's nothing super special. Okay, so that takes us through a variety of functions for the plasma. Now, as we were talking about, at the very end of this chapter, we get into what we call hemostasis, or basically how we make sure that the blood that we have stays in this cardiovascular system and doesn't leak out all over the place. Now, if you get a cut, if it's any type of a somewhat deep cut, right, something deeper than a paper cut, you will bleed. That means that you have damaged a blood vessel. when that happens, the blood is supposed to stay inside of these blood vessels, not be leaking out all over the place. Your body needs to have a way to stop that bleeding, to stop that escape of blood. And so it does so in order to maintain. Stasis means to maintain, try to keep the same. Hemo refers to blood. In order to maintain that blood inside of your system, what we're going to try to do is we're going to stop the bleeding and then heal the blood vessel. And the first three steps of this process are basically here, right? Blood vessel spasm, platelet plug formation, and then blood coagulation. And so if we have a break in a blood vessel wall, what's going to happen here is the blood's going to escape into the surrounding tissues. Now, it may leak out if it's a cut. It may be a bruise where the blood escapes into the surrounding tissue, but it's internal. But ultimately, what has to happen is, this is, a cut blood vessel is kind of like a fire hose, right? The fluid's leaking out, etc. Well, one of the ways to, if you have a hose and it's leaking, to stop the flow is to kink or bend, if you will, that hose. And in terms of a blood vessel, we're not going to bend it, but most of the blood vessels have smooth muscle along their wall, and that smooth muscle can constrict, and as it constricts, that's going to narrow the tube and reduce the flow of blood out of it. That's what we call the vascular spasm. It's actually squeezing and trying to close itself off. Now, it can't do that totally, so we have to have the other steps in the process as well. And the second step of the process is utilizing the platelets that we talked about before. And what happens is we form what's called a platelet plug. And those platelets, those cell fragments, what happens is they're usually in the inside of the blood vessel, right? So they are in contact with these epithelial cells on the inside of the blood vessel. But if you have a broken blood vessel, now they're going to come into contact with this material outside, which is collagen. This is basically connective tissue. And when it comes into contact with connective tissue, that's the stimulus that, hey, there's something wrong here. I'm not normally supposed to see that, okay? And so they will actually stick to or adhere to the collagen and start to form this platelet plug, okay? Now, the platelet plug is like putting tape on a pipe or something like that, or duct tape, It's going to seal it off for a period of time, but do remember that this blood vessel is still going to have blood trying to go into it all the time, okay? So that duct tape, that platelet plug, is not going to hold it for the long term, and we need something substantial to hold it until that blood vessel can actually repair itself. And that takes us into, instead, the clot formation or the coagulation. And the coagulation or the blood clot formation, like we said, involves the conversion of the soluble protein called fibrinogen into the insoluble form called fibrin. And like we said, the fibrin acts like, if you've ever seen like concrete, if they're doing a concrete wall or something like that, oftentimes they'll put rebar, right, those pieces of metal inside of it, and that gives it, reinforces it and gives it structure. It's kind of the same thing in terms of this platelet plug. That's the start, but we're going to start laying down these fibrin threads, these protein threads on it, and that is going to work to catch other platelets. It's going to catch red blood cells and white blood cells, and it's going to form a very solid clot instead. And so once this blood clot forms, then it's going to keep kind of reinforcing itself with more fibrin and more cells. And that is actually happening. This clotting is happening through what's called a positive feedback system, meaning that, it starts the clot and it forms more of a clot, it forms more of a clot, etc. And that's going to then form a very solid structure that can seal off that blood vessel for long enough for it to then heal. And like if you have a scab on the surface of your skin, that takes at least a few weeks or so before that scab is ready to come off, right? You take it off too early, it's going to bleed again, right? Because the healing hasn't totally occurred yet or completed. Same thing in terms of healing blood vessels on the inside. So we need that clot there for as long as possible. So after that clot forms, then we're going to be producing these fibrin fibers throughout the clot and supporting it. And ultimately, once the blood vessel has healed, we want to get rid of that clot. And there's a separate protein that becomes activated that's called plasmin. It's Plasmin. Plasmin actually dissolves the clot and gets rid of it. So then you can get back to your normal blood vessel again. Okay, so that's the clotting process or coagulation in terms of what needs to happen when you have a damaged blood vessel. But occasionally you may actually form a clot erroneously or in the wrong circumstances. And a clot that forms abnormally is called a thrombus. And so sometimes when you're on planes or in your hospital, you hear about this DVT, right? Deep vein thrombosis. If you get a thrombus, this is basically a blood clot that is formed abnormally and is now blocking off that vessel when it shouldn't be. And if you block off a large enough vessel, then the area that it was supplying with oxygen and nutrients, etc., is then going to die off potentially because it's not getting what it needs. So if it's a clot and it's stable in one particular place, we call it a thrombus. If it dislodges and it's on the move, then we call it an embolus instead. So you hear about an embolism. You can have a blood clot that is kind of gone around, and that's an embolism. If it goes to the brain and blocks a blood vessel in the brain, then we call that a stroke. If it blocks a blood vessel supplying the heart, then that would be a heart attack most likely. Technically there are different types of emboli or embolisms. You can have a gas bubble one, like if you're a scuba diver, or a fatty plaque or something like that. But a blood clot that is on the move as well is also called an embolus. All right, the last thing that we want to address having to do with the blood is blood types. And you'll see this in lab this week. But in fact, blood types, everybody knows there's different blood types. And the blood types have to do with specific characteristics about your red blood cells, but also specific characteristics about the antibodies floating in the plasma. So we've already addressed both of those. We said that, we have red blood cells floating around. We have these antibodies or globulins that are inside of the plasma as well. And so what happens here is if you get a transfusion, if you get the wrong blood type put into you, those are basically foreign cells. And if your body has foreign cells inside of it, like bacteria or like foreign blood cells, your bodily defenses, like your white blood cells, etc., and your antibodies will try to get rid of it. Okay? And one of the ways that antibodies can help to get rid of foreign red blood cells is they will actually bind to the foreign red blood cells, hold them in place, called agglutination. It basically clumps them in place, and then your big macrophages, or I'm sorry, your big phagocytes like monocytes, etc., can come around and eat up those cells. The antibodies kind of act almost like handcuffs, right? So they'll come and they'll find red blood cells that they don't recognize, and they will latch to one, and then they'll hook to another one and hook to another one and hold them all together in one particular place until the phagocytes can come along and eat them up. Okay? And that clumping process where they're holding these foreign red blood cells in place is called agglutination. And so agglutination is due to the interaction of these antibodies with these protein markers on the surface of these red blood cells, these foreign red blood cells that are called antigens. Okay? So the antigens are these proteins on the surface of these red blood cells, of the surface of your red blood cells as well as the surface of the foreign red blood cells. But the foreign red blood cells, their antigens don't match your own. And so your body recognizes it as foreign and wants to get rid of it. Okay? So the antibodies bind to the antigens. The antibodies are made in your body. Okay? They're going to bind to the antigens on these foreign red blood cells. So, as you can tell, we want to be able to get rid of foreign red blood cells, but we want to maintain and ignore, if you will, our own red blood cells. So our red blood cells have these protein antigens as well, but like we said, our bodily defenses recognize those. These antigens you can think of as like cellular name tags or jerseys or something like that. It's a name tag that says, "Hi, my name is," or "Hi, I'm on your team." If you can think of it like a football jersey, right? A football jersey there looks different between the two teams so that you can tell who to protect and who to attack. And so our red blood cells, our self red blood cells, have antigens or a jersey on them that identify them as part of our body. The foreign ones would have a different antigen that we would then recognize and try to get rid of. Okay, so let me get to the figure here. This is the text that goes along with it, but let me get to the figure to then describe to you our different types of blood. Okay? Now most people know the different types of blood, right? There's 4 different types based on the ABO antigen blood typing system. Okay? Now, like we said, these antigens are these protein markers on the surface of the cell. Now, the ABO marking system is only one of like more than 30 different ways that your blood molecules or blood cells are identified. But it's one of the major screening ways, this ABO. So, anyway, we have 4 different types, right? We've got type A blood, we've got type B blood, we've got type AB blood, and we've got type O blood. Okay? Now let's focus first on the red blood cells themselves. Okay? Now if you have type A blood, what that means is you have type A antigen on the surface of your cells. Okay? If you have type B blood, that means you have antigen of type B on the surface of your red blood cells. Notice that they're different shapes, right? The antibodies and the antigens, if they were to match up, they would have to have the same shape, like a lock and key type, a complementary shape, let's say, like a lock and a key. So, anyway, the type A blood has type A antigens on the red blood cells. Type B blood has type B antigens, which look a little bit different. Type AB blood, hey, what do you know? It has A and B antigens on its cell surface. And type O blood, which is the 4th type, the most common type, type O has no antigens of type A or type B. Okay? Alright, so now we know what it means in terms of our 4 different blood types, what that means in terms of antigens on the cells. What about antibodies floating around? Okay, remember the antibodies are looking for foreign blood types that they would then bind with, forming these clumps or agglutination. So, in the normal bloodstream, right, in your bloodstream, you want the antibodies to ignore your blood cells. So you do not want a match between the shape of the antibody and the shape of the antigen. So for instance, in type A blood, we have antibodies against type B blood, right? Because type B blood would be a foreign type of blood. And notice that the anti-B antibodies, the shape of their kind of binding areas do not match with the shape of the type A antigen, right? You can't bind these two together. They don't work. It's like putting the wrong key in the wrong lock. And that's good, right? These antibodies are guarding against type B blood because B blood would be foreign, okay? Or blood that has B-type receptors. And they don't bind with our own type A cells, so that's good. In type B blood, we already said we have type B antigens on the red blood cell surface. In terms of the antibodies, we have antibodies against cells that would have type A antigens, right? Notice again, they don't match with our own cells, so there's no problem there in terms of clumping. Now type AB blood. Type AB blood we said has both A and B antigens. So what that means is we cannot have the antibodies against A or B in with the AB blood cells. Because if we did, then we get a match, right? We would get the antibody binding with the antigen, and we would get this that you see in the lower right-hand corner, agglutination or clumping. It's not very comfortable. Feels bad to have big clumps in your blood vessels. So in type AB blood, we have no antibodies floating around. Okay? Now in type O blood, we said the red blood cells have no antigens. So that means that you can safely have both antibodies against A and antibodies against B floating around, and you'll have no issue. So that's what it means in terms of these 4 different blood types in the ABO blood typing system. You can have type A blood, type B blood, AB, or O. Okay? And normally we want the blood to look like this, right? Distinct cells, red cells, white cells, etc. We don't want it to look like this that we see on the right. So the antibodies cannot match up with the antigens in our own blood. Okay, so for blood typing, you need to know what it means for the different blood types, type A, B, AB, and O, what it means in terms of antigens and antibodies. Now, interestingly, you've probably heard that type O blood is known as the universal donor. What happens there, and this may be important to you, because if you're donating blood for extra credit or something like that for one of your classes, type O blood, they always are interested in type O blood because even though it's the most common, it's called the universal donor. And what that means is when you donate your blood, what they do is after they take the bag of blood, right, the pint of blood, they centrifuge it, they separate it, they separate the red blood cells from the white blood cells, from the platelets, from the plasma, etc. And then they take your red blood cells often, and if they're going to give it to somebody, they take those red blood cells and put them in a saline solution, an artificial plasma solution, if you will. So that way, what is being donated to the person is just the red blood cells in basically salt water, a saline solution. Okay? And that way we can make sure that you're just getting red blood cells, you're not getting any of the antibodies, you're not getting any of the weird things that could be in the plasma like hormones or waste products or anything like that. And so type O blood is known as the universal donor because these cells don't react to anything, right? Type O red blood cells have no antigens on their cell surface. So you can put them in type A blood, you can put them in type B blood, you can put them in type AB blood, and they will not react to anything. They will get along with that blood because there are no way that antibodies can bind to them because they don't have any antigens on them. Okay? So that's the universal donor is type O blood. Technically, there's also the universal recipient, type AB blood. This can receive blood from any of the other types because if you take just the cells, type A cells, type B cells, or type O cells, and you put them in here, again, you're not going to get any adverse reaction because type AB blood has no antibodies floating around. So you're not going to get any binding, etc. Okay, so that is, like we said, the ABO typing system for blood. Technically, there's also a second one that you hear of. So if you know your blood type, you're like A positive or O positive or B negative or something like that. The positive or negative is what's called the Rh factor. And the Rh factor, rather than having two antigens and antibodies, is basically just one. And so it's a question of whether you have the Rh antigen or whether you don't. Okay? If you have the surface protein, you are Rh positive. And if you don't have it, then you're negative. So Rh factor is a separate antigen-antibody system, but it's another way that we classify blood, another major way. And it was actually named because it was discovered in the rhesus monkey. And if you look at rhesus monkey, you can see rhesus starts with Rh. So that's where Rh type comes from as well. Okay? All right, so that's basically what we have to say about the blood. From here, we will then move on to the next chapter where we talk about the cardiovascular system that works to transport this all-important fluid around your body.